Sì, vediamo. E ho fatto partire
Vediamo
la
bene.
registrazione.
Ok, farò la presentazione in inglese perché c'è appunto uno studente collegato con Giuseppe che non parla italiano. Okay, so thank you for being here today, listening to this topic and to this talk. And I'm very happy to be able to present some results of my work so far.
Scusa Luisa, puoi
Sì?
parlare un po' più vicino al microfono?
Sì.
Perfetto, ora ti sento
Scusate.
bene.
So I'm very happy to present these results, and I'm very happy to be spending this period here in Lerici. So very happy to be in this place. Okay, so my topic is plastic in the biogeochemistry of the ocean. So a little bit an overview of what I'm going to talk about. So a little bit about ocean plastic, although everybody knows what the problem is. And then some introduction on plastic and marine carbon biogeochemistry and what could be possible effects of plastic together with other ocean changes, and a little bit of outlook on what could be done or what I've seen so far in my experiments and other colleagues have reported, and what we could use as a background for future studies and future efforts on this problem. So just so first reports on plastic pollution date back in the 60s, and especially they discuss about ingestion of plastic by Laysan albatross, which is a marine bird typical from the North Pacific areas. So plastic has been reported in the past, and the first reports were actually on this ingestion. And because these researchers found a lot of stranded albatross, and these were results from 1966. But oh, sorry, it doesn't go on. Okay, yes. So there are reports from the sea surface as well. So first observations at sea are the ones from the sea surface, and they date back to the 1970s. So for example, this publication from Carpenter in Science, where he and colleagues observed plastic spherules, likely polystyrene, that were floating on the surface of the Sargasso Sea. And also they estimated the number of pieces that they observed, and they were following observations still from the same year, and this from a little bit farther north, but still in the North Atlantic. And then there was this publication in 1974 where the presence of plastic particles in surface waters was discussed. And these papers were the first one that introduced a little bit the idea that plastic could also be colonized by microorganisms and could have potential effects in the future. And at that time, those effects were not fully discussed, but now we have a lot more evidence of what these authors were possibly pointing to. And yeah, this study in particular confirmed that by examining Houston trawls, that plastic did occur already at that time at the beginning of the 1970s over a wide areas of the North Atlantic Ocean. So there was another study which I found very interesting, and it was made by using the data, the available data from the continuous plankton recorder, which recorded faults that were logged between 1956 or 57, sorry, maybe there's an error here, and 2016. And this was probably the longest time series used for plastic pollution studies, and which highlight that there was an increase in the 80s and 90s, and also in the year 2000 in the concentration of plastic. And this concentration actually represents they are taken from only 30% of the total tows that were analyzed, because when the continuous plankton recorder is brought up on the surface, it's used on ships of opportunity. When it's brought up to the surface, on board of the ship, then the operator records any faults or problems that it had, and in 36% of the cases, those faults were related to plastic entanglement. So that's how we know, especially for the very previous year, that there was already plastic recorded, like in the 50s, in the surface of the North Atlantic. So plastic is, we know it's a very useful polymer, it's a very useful material, and it basically plays a key role in society. And by the 1970s, it was possibly the most used material worldwide, and it was really introduced as a low-cost, mostly single-use material that could replace other types of compounds, and therefore we start having this problem that everybody knows nowadays. So this graph here shows how plastic production and carbon emission are tightly related, although it's not, they don't follow the same path in terms of policy intervention. So policy interventions related to carbon emissions from fossil fuel combustion, there are more agreements with respect to plastic. It is to this graph comes from a paper in 2017, and now we have in 2022 we have the United Nations Environment Assembly and this establishment of the International Negotiating Committee to create a global binding instrument to tackle plastic pollution by 2024. I think that the next meeting will be in May 2023, so this year, next month in France, to discuss this legally binding instrument. And by 2024, we should have a global treaty on plastic, hopefully. So what for me is very interesting, and I've always, I've focused on my studies more or less to the ocean atmosphere interaction in terms of carbon cycling. For me, it's interesting to understand how plastic contributes to the cycling of carbon in the ocean, and through various aspects. So one aspect is, for example, the carbon dioxide emissions that are due to plastic manufacturing. And in these graphs here, taken from the global carbon budget of 2022, we see that the ocean as a sink of anthropogenic CO2 emission is increasing. And so how much of this carbon is actually due to the plastic production, and also to the amount of carbon that it's already in plastic? Just to give an overview, we have that about 4.5% of the global annual emission of carbon by anthropogenic activities, especially by oil reserves, gas reserves, and coal reserves. So by fossil carbon, it's used for it's due to plastic production. So in this sense, plastic represents already in its origin an important percentage of carbon emission to the atmosphere. And it is estimated that since the 1950s, about 8 gigatons of carbon is in the form of plastic. So where is all this plastic? There have been many estimations about the amount of carbon and plastic in the ocean. So the most famous one is the one by Jenna Jambeck, saying that there is about between 4 and 12 million metric tons of plastic that reached the ocean in 2010. And Ericsson reported that there are about 5.25 trillion of plastic particles that weighs over to 200,000 tons that are currently floating at sea. And where the majority of this mass and number of particles in the northern hemisphere, but we know that plastic can break down into smaller and smaller pieces, and that it's not all floating at the surface. So there is a lot of plastic that we don't have a clear idea of where it's going to end in the ocean. So we think that floating debris probably represent only about 1% of plastic which is introduced in marine systems. This is based on estimation like between the loads of plastic from river inflows and observation and reports of surveys at sea that in the last years have covered big areas of the oceans. But there was also an interesting study by a colleague of mine at Florida Atlantic University, Xie Zhao, who showed that particles that are below 300 micrometers are probably a critical and largely underexplored constituent of this ocean plastic inventory, especially at great depths. So what they did was sampling with large volume pumps in the South Atlantic Ocean, and they found a high concentration of polymers, especially at higher depths, and in this very small size fraction. And this was done, so these experiments that they did, this survey that they did, was counting, they had different methods, but with the large volume pumps, they also did all the digestion of the organic matter. So it's possible that some of those small particles were embedded into some biological particles. And by digestion, they were able to retrieve it and then count the amount of particles that they found. There is another study showing that coastal margins could be one of the major sinks for marine debris. And that's why we don't see all the plastic. So we don't see all the plastic that we are expecting in the ocean because a lot is going to the back shores. But on the other side, there are also studies suggesting that there could be a global biological sink where small particles are embedded into fecal pellets by the ingestion from zooplankton, for example, but they can also be embedded into marine organic aggregates like marine snow particles, and therefore they can change the buoyancy and then be exported at different depths in the water column. So that's why we don't see them floating at the surface or slightly below, and they could also be exported to the deep sea. So these pictures have been taken by a confocal laser scanning microscope, showing that plastic particles, so polystyrene spheres, can rapidly aggregate with biogenic particles like polysaccharides and proteins that are naturally dissolved in seawater. So they create these agglomerates, and this interaction of biological particles and microbial life somehow can determine the fate of those particles in the marine environment. So these aggregates that we call marine snow can have a high potential to remove particles from the surface. Although all these experiments, so these we know there's results by experiments that have been done in the lab, and not really we don't really have observations on a global scale so far, and there are models showing that, and short time experiments in control conditions. But it's interesting to see that we have already some indication. This was from 2018, but we have indications that there is this interaction of plastic with organic components. In the picture on the right, in blue, we can see polysaccharides, polysaccharide structures. In green, it's nucleic acid containing structures. In red, we have chlorophyll A, autofluorescence. And the arrows, the white arrows, indicate some fluffy stuff that could be some extracellular polymeric substances that are released by microbial organisms. And these these are the pictures taken from the same experiment that was developed by some colleagues at Geomar some years ago. Shows that there is actually colonization of plastic particles. This was polystyrene in 8 days, 12 days, and 25 days. And there are different components. We have polysaccharides, we can individuate some diatoms on the plastic surfaces, and we can find chlorophyll and also protein-like structures. And these were from a natural community taken in the Kiel Fjord in northern Germany. So these experiments led me to develop my first project on which was called Poseidon, which stood for photochemistry on the ocean surface, effects of and interactions of dissolved organic matter with microplastics. So my hypothesis was that plastic was acting as a carrying substrate, and this way could stimulate the microbial release of organic matter in dissolved and particulate forms. And here on the right side, there is a graph showing the continuum of carbon fractions in the ocean from the dissolved fraction to the particulate fraction. Basically, this is only an operational definition because it's how we sample and how we divide the two fractions of DOC and POC. But I was focusing on the dissolved fraction, especially the chromophoric dissolved organic matter. And in the particulate forms, I was focusing on marine gels and transparent exopolymer particles in particular. So on these two different size fraction of organic matter. So chromophoric dissolved organic matter is it's a high reactive fraction of the DOM pool. Could be up to 70% of the dissolved organic matter in the ocean. It absorbs UV visible light and has this exponential decrease with increasing wavelength that you can see on the left graph. And what I was very interested in is to measure the spectral slope, this parameter, which is S275 to 95. It can also be measured in different wavelength ranges. But in this range, it has been shown that it can really indicate the changes in the composition of the CDOM, because it's very difficult to understand how DOM in general is composed. So we can use the optical properties and like photobleaching, photodegradation of organic matter to understand possible processes. Like if we see that the spectral slope is increasing, it means that we can have some photobleaching processes. So the organic matter that is available has a lower molecular weight. And this happens also with microbial degradation. So anytime we have a type of degradation in the organic matter pool, in the CDOM pool in particular, we have an increase of the spectral slope. So when we have instead a decrease of the spectral slope, likewise, we have possibly the presence of organic matter, dissolved organic matter of higher molecular weight. So this could be an indication of possible fresh release by microbial processes. Marine gels, on the other hand, they are a size continuum assembly, so they are just not one standalone component, but they are made up of colloidal compounds that are found in the dissolved fraction, and they kind of bridge the dissolved and particulate pools of organic matter. And this way they can mediate the vertical flux of carbon. They represent a big proportion of the global DOC in the ocean. Like 25% of DOC is in the colloidal side fraction, and about 10 gigaton of carbon is in these self-assembled gels. And there are two major classes of these gels. This is also based on operational definitions, how we are able to stain them and recognize them. So we have transparent exopolymer particles that have a major polysaccharidic component, and we have co-mass sustainable particles that have a proteinaceous component. So we go and stain the polysaccharidic fraction and the proteinaceous fraction. And but these, I mean, EPS, all extracellular polymeric substances are also part of these gels. This is just how we can visualize them because they are usually transparent. So my project was focused on the sea surface microlayer and underlying water, which is something that I have done also during my PhD. And I wanted to understand whether the presence of plastic would have an impact on the organic matter cycling in the upper ocean. So if we think about, it's a very simplified graph, but if we could think about the sea surface microlayer as an enriched layer of organic material in general, where we can also have the accumulation of plastic and microplastics, especially at the very beginning where plastic is still floating at the surface and small particles haven't developed a biofilm yet and changed their buoyancy, so they're still at the surface. And then we have biological processes in the ocean photic zone of marine plankton growth, exudation, lysis, and release of organic matter through different processes. This organic matter accumulates in the surface of the ocean in different forms, either in the dissolved fraction, which you see here as small blue dots, and in forms of marine gels, which are these larger particles, the blue ones. And I wanted to understand whether there could be an impact on the underwater light regime and an impact on air-sea exchange of gases in this respect, because the hypothesis was that there could be an increase of production of organic matter in the presence of plastic, and this could impact upper ocean biogeochemistry somehow. The effect of photochemistry is and microbial activity in the surface is because the sea surface microlayer is exposed to high solar radiation, and it hosts a high microbial activity. So a very active microbial community, which is always recycling the material, and there could be different effects of photochemistry and microbial activity on the DOM pool in this respect. So this was done in two different ways. So first, there was a series of experiments in the lab where I used a non-axenic phytoplankton culture, and I used a filtrate that only contained heterotrophic bacteria and exudates, like this extracellular polymeric substances. I used a high concentration of plastic because I wanted to see an effect, like to see whether plastic actually could induce an effect on CDOM. I did some dark and light incubations, 5 experiments at least, and then 1 experiment focusing on sea surface and underlying water by using small tanks. and the focus was on the chromophoric dissolved organic matter. And then by using the same polystyrene particles, we did a big mesocosm experiment at the Hellenic Center for Marine Research in Crete. And in this experiment, we used 6 mesocosms of 3 cubic meters of seawater each, and they were differentiated in 3 treatments. So they had plastic in same concentration and 3 controls. And we measure CO2 in the underlying water. We measure several biogeochemical parameters, and we measured both the underlying water and the sea surface microlayer. So from the microcosm experiment, so the experiments in the lab, we saw an increase in chromophoric dissolved organic matter. So there was an enhancement of microbial production of CDOM in the presence of plastic, and this was especially by heterotrophic metabolism, so like a reworking of organic material. And there was also this change in the molecular weight of CDOM that we saw by the analysis of the spectral slope. So we went towards a high molecular weight dissolved organic matter. So possibly because there was this creation of biofilm around those particles, and this resulted at the end in a lower oxygen in the underlying water of the treatment. So where we added plastic, there was lower oxygen, lower dissolved oxygen in the system. Possibly because all these bacterial activities, so like this heterotrophic activity of reworking and this microbial release of substances around polystyrene particles, stimulated the oxygen consumption. So we also saw an accumulation of CDOM in the sea surface microlayer of the system that contained plastics. And as I said, enhanced oxygen consumption in the underlying water. So the conclusions of this set of experiments that were done over a few years was that in certain environments, like the sea surface microlayer, the presence of microplastics and active microbial communities, especially heterotrophic communities, may enhance the processing of dissolved organic matter and favor the consumption of oxygen. And so this might point to an effect on the air-sea gas exchange. It's just, this is just from a lab experiment, so we wanted to test it in a more realistic environment, and therefore that's what we saw that's why we did a mesocosm experiment. So in the mesocosm experiment, we didn't filter the seawater, we just filled the mesocosm from coastal seawater from the Sea of Crete, which had oligotrophic conditions at that time. And we saw, by analyzing the spectral slope as an indication of the molecular weight of CDOM, we saw an increase, a clear correlation of the spectral slope with the abundance of Synechococcus cells, as you can see from the graph on the left side. So when we have, so basically we had a decrease in spectral slope, which indicated the release of organic material, not the photodegradation, because it's a little bit twisted for me as well, because I mixed it up several times. But actually, this was an indication that there was the growth of Synechococcus cells and possible release of extracellular polymeric substances that was favored by the presence of microplastics. And the release of this extracellular material in the dissolved fraction that could then agglomerate and into the particulate one, but this was seen in the spectral slope indication of the molecular weight of this material, which decreased over the course of the experiment. And on the other side, we saw an increase of marine gels in both the polysaccharidic and proteinaceous fraction in the treatment. So where the plastic was present, we saw like a drawdown of microplastics in the mesocosms and an increase in marine gels in general. So probably these particles, we didn't see them any longer towards the end of the experiment, or we saw much less of those particles because they were embedded into marine gels, and therefore we could not detect them. And so this in general points to this higher release of organic material related to the presence of plastic, both in the dissolved and particulate fraction that aggregated possibly with those polystyrene particles. And this was observed in the underlying water of the mesocosms. And when we analyzed the sea surface microlayer, we saw that, okay, in the underlying water, we had an increase of organic matter with plastic, increase in these marine gels, and which is this particulate fraction of organic matter. We saw an increase in Synechococcus, so possible the Synechococcus activities was responsible for this increase in marine gels. But we also saw an accumulation of marine gels like TEP and CSP in the sea surface microlayer. So what we observed was like an enriched biofilm at the surface that kind of reduced the exchange of CO2 between the ocean and the atmosphere in those mesocosms. Because in the underlying water, we measured about 3% less of CO2 than in the controls. So to resume this experiment, which was then analyzed in two different parts. So we had those treatments and controls. The difference was in the treatments we have this high amount of polystyrene particles, and the presence of these particles stimulated the microbial release of organic material, both in the dissolved and particulate fraction. And I mean, in the particulate fraction, it was coming from the aggregation of dissolved colloids and organic carbon in general. And all this material floated up to the surface as well, where it created an enriched film. And this film reduced the exchange of CO2 between the mesocosm and the outside environment. And there were clear differences with the plastic-free controls. So overall, the results of this experiment was that there is an influence of microplastic on the dynamics of marine organic matter, because there are changes in the characteristic of the dissolved fraction, both in the underlying water and in the sea surface microlayer. There is a higher production of organic matter in general, which then goes from the dissolved fraction and aggregates into the particulate one. There are changes also in the pool of marine gels. There is a general increase in the underlying water and sea surface microlayer related to the presence of plastic. So plastic are just additional substrate for growth. Maybe they are not different from inorganic particles, but the thing is that inorganic particles have always been in the ocean, and plastic and microplastics is just increasing exponentially almost. And so the idea is that we really should pay attention to how much plastic we are introducing, because we are introducing substrates that act as a hotspot for high heterotrophic activity, because where we have a high production of organic matter, we also have a subsequent remineralization of organic matter. So this means that if at the beginning we have a system where we have possibly production of like a high primary production and therefore high oxygen production in that period of time, but we have also an accumulation of a lot of organics of the surface that block the exchange of gases with the atmosphere, the subsequent remineralization of this organic material produced by heterotrophic processes will develop more CO2 and will possibly lead to oxygen deficiency in certain environments. So it's a balance that we have to, in my opinion, we should take into consideration. And the other thing is that we know that plastic actually carries carbon. So there are many ways in which plastic interacts with the carbon cycling and carbon biogeochemistry, because there is possibly this double effect. So there could be a concentration of nutrients on solid surfaces, and therefore if we have a plastic substrate, a plastic particle, this concentration of nutrients can stimulate bacterial respiration. So plastic is just not, it's not an inert substrate. It can really carry carbon through processes, not just because plastic is carbon, so can leach carbon, but also because all this amount of microorganisms and activity that concentrate on these particles, and this hotspot could be problematic in certain areas because they actually could lead to areas where we have an oxygen deficiency. It has been shown also that there might be a selective attachment of plastic, so there might be a competition in time and between different species to be able to stick on the plastic surfaces, and some leachates of plastic can impair the growth and oxygen production of Prochlorococcus. So plastic in general can have an effect on microbial dynamics. And then all these with these previous experiments in mind, and this idea on the carrying capacity of carbon that plastic has, I wanted to understand whether plastic could have an effect on the carbon pump in the ocean. And this is more or less a topic of my project right now, and where I'm looking also at microbes more in depth. But these were results from an experiments that we did with sediment traps in the North Atlantic, where we found a clear correlation of marine gels and plastic. So marine gels and marine snow and plastic as a removal mechanism of plastic from the sea surface. So we had 2 deployments at different depths up to 600 meters, and we measured the plastic flux in situ, because one idea was the mesocosms that we saw this drawdown of plastic related to the concentration of gels, but this was an experiment. We wanted to see that in real setting, and so that's why we used the sediment traps. And we saw actually a clear correlation between the flux of carbon, which is contained in the transparent exopolymer particles, which is this polysaccharidic type of marine gel, and the flux of, for example, polyethylene terephthalate and other polymers. We also analyzed one particle that we could retrieve large enough for confocal laser scanning microscope, and we could see some different bacteria on the surface, but also unspecified fluorescent material that could be individuated as EPS, so extracellular polymeric substances, and this was a particle retrieved at 300 meters. So there is an activity, and this points to this idea of hotspot for remineralization of organic matter, even at depth. And what was interesting to see was that the plastic that was the carbon bound in plastic could be up to 3.8% of the total flux of particulate organic carbon in this experiment. This was in the North Atlantic Ocean, and Xie, my colleague who was working at Florida Atlantic University, he found similar ratios of carbon plastic to POC in the North Pacific Gyre, and these are recent results that he published. So it could be that the plastic which is in the POC that we measure, the carbon is like a high proportion of carbon from POC is actually related to plastic. In our experiment here, it was more than the marine gels carbon. So plastic has a certain share of carbon in the global carbon, sinking carbon in the ocean. So maybe it can have an effect on the biological carbon pump. So to resume, there are I published this 2 years ago. It's like a short review on what I think would be several processes on how plastic can act on the marine carbon biogeochemistry. So when plastic is staying at the surface, it can stimulate the production of organic material in the sea surface microlayer in particular, and this can promote a reduction in the intensity of light and also in primary production in the euphotic zone. And on the other side, we can also have an impaired production of oxygen and carbon dioxide related to small hotspots of activity, because if we have if plastic acts negatively on the performance of phytoplankton, for example, but supports microbial heterotrophic metabolism, then we have an impaired production of oxygen and carbon dioxide already in the water. The accumulation of organic material related to plastic, related to the presence of plastic at the air-sea interface, can create a barrier for air-sea gas exchange. And then these the plastic sinking, the plastic associated with the sinking particles might represent a great proportion of the organic carbon which is sinking at depth. And also on these aggregates that are sinking, we can have an accumulation of nutrients that can stimulate microbial heterotrophic growth and respiration. So maybe this is not critical right now with the present concentration of plastic in the ocean, but could be relevant in the future if plastic production continues to increase, and also to be released improperly into the environment. And I found very interesting these studies by Christina. She's a very good friend of mine, and she did this study on the carbon which is leaching from plastic, and in 2 different papers she found that on one side plastic can leach carbon, and this further stimulates the heterotrophic microbial activity. And with other experiments that she performed more recently, she also saw that the plastic leaching of carbon can reduce pH in the ocean. So she did it in small experiments, but this could be something worth investigating on a larger scale. And then we also know by other authors that on a global scale we have a decline in global oxygen content, and Karin Vale, she modeled that in areas where nutrients are not depleted, but there is a high concentration of plastic, there might be a preferential grazing of zooplankton on plastic rather than phytoplankton, and this could accelerate global loss of oxygen through processes of initial high production of organic matter, so initial high primary production, and subsequent high degradation of this material that leads to oxygen deficiency. And this is what she saw in the model. So in areas that are not nutrient limited, that is the graph that you see on the lower left hand of the picture, the presence of plastic and the presence of nutrients might lead to higher oxygen consumption with respect to other scenarios. And she calculated that this additional remineralization due to the presence of plastic could decrease water column oxygen by as much as 10% in the North Pacific. So this is a model, but it could be very interesting to be evaluated as well on a global scale, or at least in larger scenarios. There's also a potential impact of plastic in the carbon and cycles of nutrients. This also needs further study. We know that plastic attached heterotrophic bacteria have very high cell-specific activity, which can be much higher than the free living fraction of bacteria. And these results were done from experiments of 45 days, but we would need longer incubation time to really understand potential global interaction on a larger scale, to really understand how all these processes affect the carbon cycling through the microbial loop and through the biological carbon pump in the ocean in general. We could start from specific areas, specific like enclosed sea or coastal areas with simulations to understand, to see how the impacts could be strong in those environments, to see whether there is an effect or not. And I found also this review very interesting and kind of summarizing more or less what we have discussed about so far. So it's really unknown what the effects of plastic could be on a larger scale in terms of time scales. There could be effects on decades that are relevant for example, by cycling of carbon and nutrients, dissolve oxygen, more from the physical point of view through different interaction, for example, with the interaction with zooplankton, with the interaction with primary producer, with the interaction of phytoplankton, so reduction in phytoplankton performance, rather than biogeochemical carbon cycling alone, but still we would need more studies, either modeling or simulations to show these impacts. So just to summarize, I think in general, by what I've seen in my experiments and what my colleagues have seen, and these more or less are all colleagues, and we we exchange ideas and we work closly together between most of the papers that and the results that I show you, that we are working on this increase of organic matter related to plastic. And we know that there might be an important amount of cell-bound carbon on plastic. So we have hotspots for organic matter remineralization, heterotrophic respiration, but also carbon that is carried by plastic in different areas. We think that plastic can lead to altered SC gas exchange dynamics and can possibly lead to oxygen deficiency zones because of higher remineralization of organic material and an impediment to the escape of gases through the surface when we have a high accumulation of organics. Plastic can represent up to 4% of sinking particulate organic carbon, but this number could increase if with increase in plastic concentration in the ocean. And plastic leaches carbon, and this can increase ocean acidification. And we know that plastic leachate can impair phytoplankton growth. There could be, as I said, physical mechanisms that could have a larger influence than chemical ones on short time scales, or decades to a century. Maybe chemical mechanisms might prevail on centennial or millennial timescales, but there is a large uncertainty about that. So in general, we should I suggest we should focus on those aspects and those interactions to be able to really understand plastic as a global, as a seawater component possibly, and which has maybe its own biogeochemical cycle at this point. And with this, I would like to thank my colleagues who I have been working on with, who have been working with me on those two projects in these years. And I also would like to thank Alessandra, Giuseppe, and Stefano for hosting me here at ISMAR, and you for your attention.